Well, I'll start talking, kind of give the intro, and then you'll hear it all again in the video walking through our lab, and then we can open up for questions. But just in advance, kind of how the webinar works, I'm going to show a video that we pre-filmed. You'll see me with my mask on, so hopefully you can understand me while I'm walking through the lab, and then we'll open up for questions. I'm going to turn the lab video on here right now. But just to introduce myself, my name is Vlaisavljevich. Again, I feel bad. I'm going to be saying this on the video as well you're about to see. But I'm in the Department of Biomedical Engineering, and my lab is a focus ultrasound lab. So I'll stop there, other than to say we're in the Department of Biomedical Engineering and the Blacksburg campus in Kelly Hall here. So I'm going to show the lab tour now, walk through, and then we'll kind of take questions from there. And I appreciate you all joining, and I look forward to kind of chatting with you after I share this video. Well, hello, everybody. My name is Vlaisavljevich. I'm an assistant professor in the Department of Biomedical Engineering and Mechanics. So today we're going to take you through our lab, which is down here. First thing I wanted to mention is you can see, these are some of it's basically a joint lab space. So if we walk down, you'll see, this is something we have for biomedical engineering labs. It's a highly collaborative field, a lot of our labs working together. So if you kind of come down, each faculty has their different lab space, and you can see down on the end is ours. So what you can actually see a bubble cloud up on the wall. Connor and Maggie here will show you some examples in a second. But basically, what our lab is we're a focus ultrasound lab. So this involves basically, for most applications, it's in cancer right now, as well as some other clinical applications. But basically, for those of you who are familiar with ultrasound imaging uses really low pressure sound waves that are put into the body and reflected back to create an image. And that's how we do medical diagnosis using ultrasound. In contrast, and this is an unpopulated transducer here, but you can see this is one of the prototype scaffolds we make for our devices, which is, you can see how it's focused down, like a curve, like a lens shape. And what this does is we use very high pressure sound waves that are focused down to a single point in the body. And this is to induce some bio effects. So most of our work is focused on generating these bubbles, which you can see up on the screen here in a second, or she'll show you there. But basically, we generate these cavitation bubble clouds that are really localized at a focal point in the tissue. These bubbles expand and collapse and basically destroy the tissue into a liquid debris. So basically, one of the big applications for this is non-invasive cancer surgery. So in 2019, with our partners at Histosonics, which is a company developing this technology for the clinic, as well as our clinical teams in Wisconsin, Michigan, and Barcelona, where the trial is run, we did our first human trial using histotripsy for the treatment of liver cancer. Our lab's now developing this for a wide range of other applications, including pancreatic cancer, soft tissue tumors, bone tumors, brain tumors, and on. So the way this works here is if you kind of come around here, you can see this is one of our benchtop setup prototype focus ultrasound transducer that we've developed in the lab. It has individual ultrasound elements focused down to a single point. And then when you actually run the treatment, right at the focal point, we generate these really quick bubbles. And I don't know if you'll be able to hear them on the video, but we can change the rate in a second so you can see it directly. So if you look up on the screen here, this is showing a high-speed camera image of what those bubbles look like at the focus. And you can see, so it's a really localized, well-defined bubble cloud. And if we come back over to the tank here, Connor can increase the PRF, or the repetition rate, and then you should be actually able to see right at the focus. You can hear it now. I don't know if you'll be able to see. Yeah, you should be able to see that in the tank, kind of that flashing there. And that's basically, you're getting all these bubbles at one point in the tissue. Let me shut it down now, I think. And that's what's going to be destroying the tumors when we treat. So that's most of our lab. You can see this is our benchtop setup. We use a lot of these optical tables and high-speed cameras or ultrasound imaging feedback. We have other experimental setups with our partners in the School of Veterinary Medicine, where we do small and large animal experiments. So these include tumor studies in induced tumor models in mice, as well as large animal studies in pigs, really working on device development, showing proof of concept that we can do this in a live animal model. We're also running clinical trials on veterinary patients, so dogs that have soft tissue tumors as well as osteosarcoma tumors. And we're really interested not only in human medicine, but veterinary medicine as well, in having non-invasive surgical options for treating tumors in dogs. I can kind of walk you through the rest of the lab. It's very similar benchtop setup. This is more of our in vitro work that we have wet lab space, benchtop. We do a lot. If you look to the left here, so electronics equipment. So a lot of device prototyping. So we make everything in-house, including the electronic driving systems, amplifiers, our focused ultrasound transducers themselves. In our other lab at our animal experiments, we have more of like a robotic micropositioner that's closer to the clinical setup that will eventually be used. And then in addition to histotripsy, which is destroying soft tissue tumors, we also have other projects that use focused ultrasound for non-invasive neuromodulation, so actually activating regions of the brain with focused ultrasound. We have some biomaterials projects, controlled drug delivery work, and finally we're working with using focused ultrasound to actually extract DNA from timber and plant tissues as part of an environmental conservation project to say more rapid field deployable tests to be able to diagnose illegal timber trading, as well as being able to actually on site be able to identify what type of plant and timber tissue we're looking at. So if I walk through the rest of the lab, it's going to be very similar. You're going to see a lot of device work, setups. This is a high-frequency ultrasound imaging system that was built by a company called Daxonics and a collaborator of ours in Halifax, Nova Scotia. We work very closely with Dr. Jeremy Brown, custom developed this, and now we're building, in partnership with him, these endoscopic histotripsy systems that utilize his technology for this high-frequency imaging. You can see a lot of the other prototype devices. So these are prototype lenses that we make for our ultrasound transducers, and you can see a whole lot of different setups and a lot of 3D printing work. A lot of we're getting more into injection molding as well. So very device-focused biomedical technologies in our lab. You can see one of many different types of 3D printers that we have going on here in the lab. This is just a basic one for little lab setups, and then we have some nicer ones over in some shared space for different types of building the actual devices. In addition to this, we have multiple cell culture hoods. So those of you who are interested in more biologic studies, so this is for our DNA-based work, as well as we do a lot of cell-based studies looking at the effects of histotripsy or other focused ultrasound, including our nanoparticle-guided histotripsy projects, looking at what's the response of cells to this therapy, what's the activation of the immune response, and other things. You can see here, I mentioned timber, so even though we're a biomedical lab, we have our timber samples that we're trying to get DNA out of. And so then we have kind of separate little stations here, and then if we go in here, this is our cell culture hood. We won't interrupt Maggie as she's doing experiments here, but so Maggie has worked in the lab as an undergraduate student for what, 3 years now?
Yeah, coming up on 2 years, I think.
Okay.
I started in the summer of 2018.
Okay, great.
Undergraduate student, yeah.
And she's graduating this year and going on to, sadly, leaving the lab and going on to bigger and better things. So, but she's working on a project to develop nanoparticle-guided histotripsy for the treatment of metastatic breast cancer right now. So this is more of our sterile cell culture facilities here. And then around this corner, we won't go too much into here because it's a little more crowded, but you can see a lot of our custom designed amplifiers that we developed in the lab, along with our collaborators that are more electrical engineering. So my former group at the University of Michigan, Dr. Tim Paul did a lot of these initial designs, and we've replicated them here and continue to collaborate closely, not only with partners at Virginia Tech, but really around the world. So a lot of prototyping space and different, we're going to put some 3D printers back here too, but mostly device and tissue prep work for our various experiments. So I think that's pretty much it. Anybody's interested in learning more about focused ultrasound or just biomedical engineering in general, throughout the department, easy to find online, and feel free to send me an email, and we'd be more than happy to talk to you about the work we're doing and others in our department and across VT, and hopefully we'll see many of you here in the coming years. Thank you. All right. Well, thank you for those of you who are here listening to that. Hopefully, you didn't mind listening to me talking through the math there and trying to adjust it as I went. But as I mentioned, my lab is a focused ultrasound lab. We have mostly histotripsy, which I talked about, which is the cancer ablation, and there's a wide range of applications and features. I'd be happy to talk, about that, as well as some of our other projects in non-invasive neuromodulation, DNA extraction, some of the biomaterials work, or if you're interested more just in general in the department, biomedical engineering, the program, anything. Okay, so Lauren asks, how early are students able to get involved in lab work? So that's a good question. I think it really depends on, I'll speak more generally and then talk more about my lab. But I think it really depends on, how soon really the student knows what type of research they may want to be involved with, as well as what kind of major they're in. So I think, for instance, some students, kind of takes a semester or two to kind of, fully define their major, look around, meet different faculty and labs. And a lot of people end up starting, junior or senior year as they start thinking and really defining, okay, I know specifically what type of work I'm on. But I don't think that has to be the case. I've had a lot of students, at least multiple students I've had start working in our research lab in their first semester after coming to Virginia Tech. And I've had others that have started really late, I think even, some who are the final semester. Usually the types of projects the students work on in our lab vary. So it tends to be a process that the longer you're in the lab, the more you end up taking on independent projects that you're leading, where first when you join the lab, there's a lot of training, skill development, working with other students. We're a highly collaborative research program in our lab, and we work very closely with other faculty, not only in biomedical engineering, but clinicians at Carillion and other places. A lot of faculty in the School of Veterinary Medicine on our various projects. I have a lot of students who kind of move between labs. And for undergraduate students, typically they'll join the lab, and then as they kind of develop that skill set, they'll be leading projects by the time they end up graduating and leaving our lab. So it really varies, and I think the biggest thing is just reaching out to faculty early on and connecting. I have a lot of students who are excited to get started with research or just anything that's involved with the areas they're passionate about early on, and I think that's a really big benefit to them, to kind of having multiple years working in the same lab or different labs as they kind of see what's the best fit for them. So another question, what is your favorite project that you've worked on in your lab? So that's difficult because then some people from my lab will see this, and I don't want to pick winners and losers, because I obviously like all of our project. I would say they all have their own rewarding aspects. So I'd say the most personal project to me, which was one of the main projects for my graduate school work that continued on here, was developing histotripsy for the treatment of liver cancer. And the reason that's personal to me is my mom died of liver cancer when I was 4. And so coming into grad school, having the opportunity to work on a brand new technology to non-invasively treat that disease was really rewarding. And also it's, been rewarding because it's such a long process, going right from, early concept phase, benchtop testing of a new technology, through in vitro studies, small and large animal in vivo studies. And then I actually worked for a startup company that formed out of my former lab, Histosonics, for a couple of years, building our first clinical prototype system. And then that, once I joined Virginia Tech, we were able to use that system in partnership with the company and the clinical team I work with in Barcelona to do the first ever human clinical trial. So that was a real full circle moment, seeing, okay, a disease I really care about, improving the treatments for, actually getting a new technology into the clinic. And now Histosonics is doing their first US-based clinical trial currently, and hopefully we'll now see this histotripsy come first for liver cancer, but then for a lot of other indications come into the clinic and be used widely and really help a lot of patients that don't have good treatment options, a lot of side effects with current options. So and really expanding the patient population. So I'm really excited about that. That was extremely rewarding. But at the same time, I love some of our projects that are real basic science. So I've done some studies, so histotripsy uses these cavitation bubbles to non-thermally destroy cells. So developing little cell bubble systems where we can see mechanistically, basically how to nucleate bubbles in water. What are the properties of water in tissue that impacts the therapy? How do these bubbles kind of eat away at the tissue? So that's really enjoyable. I've had a lot of fun recently with this DNA extraction project, where actually a student who took over my undergraduate research project when I was an undergraduate student. His name's Hal Holmes. He went to grad school and then came back and joined our lab and came into our lab as a postdoc, who's now still working with us. He works at a nonprofit now, but still collaborates with us. He came in and asked, can you use histotripsy, which is our cancer therapy, to treat trees? And more specifically, can we erode timber to release DNA that's trapped? Because it's really hard to do genetic testing of plant and timber tissues. So that's been a lot of fun because it's we have wood and timber samples in our lab now, and we're kind of moving into this whole different area. So that's kind of a favorite for a different reason. But really, all the work we're doing, the non-invasive neuromodulation is really interesting. Looking at treating things like treatment-resistant depression, other neurological disorders, getting into things like epilepsy, and that's really basically activating or inhibiting regions of the brain. So there's a lot of technology development and mechanistic, bioeffect studies that we're interested in doing there. So I'm excited about all of it. That's why I sound so caffeinated. So good questions, though. Hard one to answer. Okay, next question. Have you ever combined another field of engineering with biomedical? So yeah, that's a great question. And I would say all the work we do, and biomedical engineering in general is highly interdisciplinary and really integrates all other or most other areas of engineering already as a field. But the answer is yes. Every single project we have is highly integrated with other areas of engineering. So and I actually look on a graduate student level to hire biomedical engineers as well as mechanical and electrical engineers. And most of the students who work in my lab, because our biomedical engineering program for undergraduate students is fairly new, most of our undergraduate students come from mechanical engineering, electrical engineering, BSc, and some of the others as well, as more of the biology fields. But our work, all of our systems develop really use a lot of electrical engineering because we custom design, really high-powered amplifiers for driving focused ultrasound systems. A lot of mechanical engineering for 3D printing, designing, the mechanical structures of our CAD models and doing, more mechanical models of acoustic propagation. A lot of bio involved too. I know that's not engineering, but working with biologists. We work really closely with immunologists because recently we've seen histotripsy can actually activate an anti-tumor immune response. So trying to study that thoroughly to understand, okay, we're treating one tumor and all of a sudden we're seeing other tumors decrease in size because we're activating this immune response. What are the mechanisms there? So that's highly integrated into what we're doing. Trying to think if there's other types of engineering. There's a lot of professors or other research in AOE, so aerospace and ocean engineering, who I work with more on cavitation physics, just because there's overlap with cavitations not only used for killing tumors, but it's relevant in ships and propellers and a lot of other areas. So there's a lot of collaboration there. But yeah, definitely all of our projects and all of our work is highly integrated with multiple types of engineering that are either all within our lab expertise or collaborative projects that involve multiple labs across campus or across the world. How do you pick the students in your lab? So for the most part, I would say I try to look for students who are, well, first and foremost, who are passionate about the type of work we're doing and undergraduate research in general, or undergraduate and graduate research. So I look for people, most of the work we do is really translationally minded. So we do a lot of basic physics as well, a lot of pure engineering, but it's all moving towards, and that's a big focus of the way I've structured the lab, is focusing on trying not to just do the research for the research sake, but get something into the clinic. So I think that really is attractive to a lot of people who have, kind of the shared passion that I do about working in this kind of bench to bedside medical device translational science approach. So the first thing is I want to make sure that any students who usually reach out to me looking for positions, that once they hear about the work we're doing and I hear about what they're interested in their career, that this actually fits their interests, right? So for instance, we do some computational work, but we're not a highly computational lab. So I'll have students sometimes who are really interested in computational work that I'll maybe send them to another lab that would be a better fit. So first and foremost, I pick students based on the fit. Then looking at obviously the needs of the project. So for instance, our nanoparticle guided hysterectomy project is a great example of a project that needs multiple people from different expertise. So I need engineers, both mechanical, electrical, biomedical, to build systems, build the technology, run a lot of the experiments. But we also need chemistry to design our nanoparticles, some of the animal work, veterinary. So we really need people from different backgrounds. So often there'll be a gap. So I'll have, let's say, a graduate student who's more on the engineering side, and I'll bring in an undergraduate student who can help with cell culture, or who's interested in more of the bio side or the chemistry side. So it's a bit of a fit on the need of the lab and the specific projects as well. And then from there, I kind of look at, obviously, resumes, CVs. I view research as really being a privilege. So the big focus of this is to make sure that any student who joins our lab, it's not going to detract from their coursework and the other things they're working on. So making sure, if they're taking 18 credits and really struggling, now maybe this isn't the time to join the lab. And not to say you can't have a lot of credits and do research. You definitely can. I have students who do that, but try to make sure that we talk at the beginning and make sure it's a good fit, and that the student can dedicate the amount of time to really be productive in the lab without detracting from what they're doing. Hopefully that mostly answers it. As far as the process, I would say for 99% of the time, it's students just directly email me that they're interested in research, and then if I have something available. And it's been tough this year, obviously, because our lab's now so big, and with all the COVID regulations, it's been hard to add more students, but we're still, we have a lot of students in the lab right now, and we're continuing to grow as we move forward. But it's typically students will email me, or they'll talk to one of my current students and reach out to me that way. And then every once in a while, especially when I first joined Virginia Tech, I'd put out more ads looking for students when I have a big need, but that rarely happens now, because there's enough, I would say, enough interest or people hear about our type of work and have reached out to me. And that tends to be a good way too, because you get people who are interested in this type of work. So how long do students typically spend in the lab? So if you can clarify, or I can answer it both ways, are you talking about kind of like the duration in months and years, or in weekly or hourly times in the lab? I guess I can maybe answer just both of those, but okay, weekly. So it really varies between projects and between students, and obviously from looking at undergrad versus graduate. As grad students are in the lab a lot because it's a big focus of what they're doing, and as they get done with their coursework, it's pretty much their full-time position is doing research, so they're in the lab a lot. With undergraduate students, it really is a bit of a balance. I would say when I have undergraduate students who are doing research for credit, our typical guideline is the number of credits. For every credit, it corresponds to a minimum of about 3 hours per week in the lab. So a student who's taking research for credit for 3 credits is going to spend 9 or 10 hours a week in the lab. And obviously I'm fairly flexible as that oscillates week to week with exams and other things. And, I do the same thing. We have 8 medical students from, the Virginia Tech Carilion School of Medicine that are currently working with us. And they're the same way. Their schedules really fluctuate based on what course they're on, what research block. So there's some flexibility and oscillation there. But typically I think that, for students doing research for credits, and that, 8 to 10 hours a week. And then I have a lot, most of the students in the lab are on, paid wage positions that are, just more like a undergraduate research paid positions. So they're, I would say, again, it varies probably within that same range. Sometimes if it's a busy week and they have other things going on, or there's not a lot of need for the project, if they're helping another student versus leading their own project, it may be as low as 4 or 5 hours a week. But there are weeks where we have our undergraduate students who maybe are in 20, 30 hours on top of what they're doing, that they're just kind of going above and beyond, or we have a big set of experiments, and so that's definitely not something that's forced or expected. But as students kind of take on more of lead roles on the project, it tends to grow into that type of more of a workload, but hopefully more of a independently led into that workload, where they're excited to do more of their work, and obviously, again, making sure it always fits into their schedule and it's not detracting from their other progress that they need to make. So are students assigned roles for the entirety of a lab, or do the positions and roles vary day to day? So I think this is so it again, it's if you think about it in terms of like the way our research lab works, usually students are first assigned, first they're going to get their training. So once kind of the general lab-based training is over, they're going to start on being kind of assigned more of a project. And that project will include different roles. So for instance, I mentioned the nanoparticle project. I have a couple undergraduate students who are mostly doing cell work. So that'll include passaging cells, doing experiments where we're, developing 3D cell culture models, doing imaging work, but it's all kind of around this cell culture. So their work's fairly consistent on that project where their work is cell culture work. But as far as a student who's kind of, let's say, leading the whole project or working on various aspects of the project, they may spend, let's say it's a project where we're developing a new histotripsy transducer for one of our veterinary applications. So we have some projects that are treating bone tumors in dogs through a veterinary clinical trial right now. So one of the way that project kind of evolved is we started by having to develop a histotripsy transducer. So I had a couple students who were, looking at, basically the user requirements of a transducer, what's the focal distance, size of the transducer, the pressure we need to output, included some modeling and building up CAD models of what a device would look like. Then we basically 3D printed those and custom built the amplifier. So there were a couple weeks or longer where they're actually building devices, doing a lot of like the actual hardware work. And then from there, once we had the device, they're doing more benchtop type experiments in water tanks, testing whether on excised tissue samples we can ablate them. And more recently now, since we've had that device fully built, they're actually working, a couple of those students, on actually with our veterinarians in the vet hospital treating these tumors in dogs. So in that sense, it varies over the course of a project. And between projects, even, some students will start and join the lab, and then they'll switch to a different project, either because their interests change or the needs of the lab change. So there is a bit of fluctuation, but for the most part, it's not something where they're working on one task one day and another task another day. There is continuity, but more thinking in terms of what the project is, less in terms of kind of assigned roles. It's not really, I don't really take students where they're just doing assigned kind of task-based stuff. They're even early on, when they're more in that role of helping with a specific part of the project, I always want them to be integrated into that whole project and thinking about every aspect of the project, not just kind of you're not just assisting with the lab in the sense of being kind of just doing manual labor. It's more they're leading the research project or closely collaborating with whoever the student or staff member who is leading that project. That's definitely a good question, because it's research can be hard and there can be some tediousness to it, and you definitely have to keep after it. But it's I think overall, over the course of months and years, it's definitely, at least the type of work we're doing, shouldn't get boring. So definitely work, but should be exciting. You should get involved with different aspects of the project and get to work with a lot of the different tools within reason and interest and where the projects are. So next question. This is a good one. No, they've all been good. Another good one. Have you gotten to interact with the people who are affected by the issues you're trying to solve? So yeah, it's interesting because obviously I'm biomedical engineering, but I'm not a clinician, so I'm not on the front line. So I work a lot with the clinicians who work with directly with people who have various diseases. So I don't directly interact, for instance, with a lot of cancer patients, although I did go to Barcelona and meet and actually was there for one of our first histotripsy liver cancer patients. So that was really interesting, and interviewing the woman who got the treatment, who was really excited to for a phase 1 trial. It's you're not at that stage. It's really an initial safety study, so you're not telling them you're going to be curative. They're really late stage patients, so it's really sad to see. But she was so excited, not only for the potential of this therapy, but just to be a part of something that could help so many other people. So that was really inspiring. And I try to stay as close as I can and do as many clinical rotations as I can, so that I'm not detached. I'm not designing something that doesn't really know what the clinical, because that happens a lot in the field, because you think, okay, it's so easy to ignore things like how long the treatment can last. So, for instance, in my view, if we can completely ablate your tumor safer and more effectively, that's sufficient in my head often. But now I realize, okay, the way the clinical practice works, if that's going to take a 3-hour window, maybe they wouldn't use it. So we might need to be safer, more effective, and more efficient on our treatment time. So you really understand some of the practical limitations, as well as obviously what it takes to actually get to the clinic through FDA regulations, et cetera. So I try to stay as close as I can, but for most of the applications, I haven't been. So for instance, I'm not, I work with on our new project we have for neuromodulation for depression. I work with Dr. Sarah Clinton in the School of Neuroscience, and Alon Kerman, who's a psychiatrist. So he works very closely with a variety of patient populations who could benefit from this, but I haven't interacted with them directly. I will say we've been with our veterinary clinical trials. It's been we've been interacting with the dogs directly, obviously, when they come in, and that's it's really sad to see on some of these. So the trial, we haven't started our brain tumor trial. We're about to start that one, but we're treating soft tissue sarcomas. Survival's pretty good for them. These are treat and resect studies. So we're doing a treatment, and then we're still removing the tumor and looking at it, so that, looking at immune modulation. But for some of the bone tumors, with the osteosarcoma tumors is the name of these primary brain tumors. That one's really sad to see in the dogs. I mean, the frontline therapy for these is still, complete limb amputation, and some of these, the pain that's underrated. I've seen human patients with bone tumors and how painful and debilitating they are. And so having not only a non-invasive approach, but our non-thermal therapy can be tissue selective. So we think we'll be able to hopefully make this into a limb sparing, so you don't need to get amputated. So anytime I do get the opportunity to be exposed to people affected by them, it's obviously, makes the work so much easier to plow through and keep working hard. But I get it more indirectly through the clinicians that tell you the stories, and probably better. I couldn't be a clinician. It's really hard to do what they do, especially working with some of the nurses as well, who are caring for these patients. And so one of the projects I didn't mention, but we started working on basically the non-invasive ablation of biofilms for the treatment of urinary tract infections. You see these kind of systemic infections, and nurses who have to work with people, and they're really frustrating that there's no better treatment options. So when I hear them say that, it's a reminder of, one, being very thankful that we have these type of people taking care of us, and also that we need to get them better tools as soon as possible, and make sure we're focused not on just doing things that are fun and interesting, but are hopefully impactful long term. And I'll just mention, as the next question comes in here, that my email is ELIV, so eliv@vt.edu. You can look me up online. So I'm always happy to follow up with any of you if you want to talk sometime, or just email me if you have questions, or if you need me to put you in contact with other people. So definitely don't hesitate or be shy about that. So the next question: is there a resource where we can read and learn more about the biomedical labs conducted at VT? So I think just our general biomedical engineering website is a good place to start, because it links to the different faculty pages, and then you can link to the faculty. And like, I have a web page that you can get if you see me on the biomedical engineering department, or BEAM, so biomedical engineering and mechanics. You'll be able to click on each of us, and you'll see the different research that's done in our lab. It'll bring you to our specific lab pages, and then there'll also be some general information about our more general biomedical labs that we use for teaching, et cetera. You should also look at some of the other non-BEAM. So we're biomedical engineering and mechanics, but there's also the Fralin Biomedical Research Institute up in Roanoke, and the Virginia Tech Carilion School of Medicine. So there's a lot of biomedical research going on at those labs up there, including my own. Like, we work closely with different labs up there and partners. I have medical students work in our lab who are primarily in Roanoke and go back and forth. So there's a lot of labs that if you look up the Fralin Biomedical Research Institute, FBRI, you can kind of link to those type of other biomedical labs. And if you have other questions, feel free to email me, and I can try to point you in the right direction. one thing about biomedical engineering, it's not only, or biomedical research, it's definitely not only housed in one department or one place. It's really all over campus. I mean, there's a lot of biomedical research going on through our School of Neuroscience and other departments. Mechanical engineering, there'll be mechanical engineering professors who run labs that have, let's say, a prosthetics component, for instance. So I would definitely say it it's all around VT, but as a starting centralized point, and a lot of those faculty will have affiliated positions. So if you start with like the FBRI website or the Beam website, that'll probably get you the closest to what you want as far as being able to access most of the biomedical research that's going on. And again, if you have a specific interest area, let me know. That's easier to point you in that direction. So if you come to me and say, send me an email and say, I'm really interested in who's doing cancer research, and that goes with the different people within our department. We have great support if you look at both at undergrad, graduate level, whether it's research or teaching. If you reach out to people in our department, they'll be able to say, what are you interested in, and connect you with the right labs that you might want to start looking more into. And I should also say, if any of you are interested ever in meeting with any of the students who work in my lab or others, they're always happy to jump on Zoom meetings or exchange emails or talk to you about their experiences. And they can answer things from a different perspective than me, of course, and having navigated their various routes into research and where they're going. I think it's really interesting to see, especially in biomedical, there's so many different directions that our students are going. So I have multiple students, both undergraduate and a PhD student, who are going to medical school after leaving our lab, which is very common for biomedical engineering students in biomedical engineering labs. A lot going into different industry device companies, a couple working more in national labs or working at NIH, for instance. So there's different routes, and that is something that I try to spend a lot of time when I have students join the lab to get a feel for where their interests are. So if a student's very interested in going into industry and working on more hardcore device work, I make sure I pair them on a project that they're going to get hands-on experience building devices, building amplifiers, transducers. Whereas some of our medical students, or students who maybe are more interested in cell-based, biological-based labs, they tend to navigate more towards our cell culture work, more of the, I wouldn't say basic science, but more of the in vitro type studies that we're doing. And similarly, I've had maybe two or three. I've had a couple students who have gone on to vet school or are currently interested in going to vet school. And for them, we try to make sure they're getting some experience with our animal experiments, and particularly with our veterinary clinical trials, because it really fits, the experience they need for their career. So it's definitely not only about what the lab needs or even just about the research, but it's, I'm really passionate about making sure that anybody who's working with us has, that it fits in their story as well and where they want to go. And if our lab's not the right fit for it, we make sure we find, somewhere else they can talk to try to find a place that they're going to feel, that they really fit and are really going to be able to learn what they want and have fun doing this research. I mean, I think research should be hard work but enjoyable, and hopefully that's the experience all of you have in the future or have already had. So this is one I probably don't know, so I'm going to have to put it to somebody else. But the question is, what are the most common minors that biomedical engineering majors choose? Do they tend to stick with STEM minors, or students gravitate towards other fields as well? So I guess I can't really give you the right answer on the stats of it, because I don't know those numbers. So I can just kind of guess a little bit. And I will say this is not for Virginia Tech, because our biomedical engineering major is brand new still. So we're still in our first few cohorts. So my experience has been, because we didn't originally have a biomedical engineering major, that most of the students who work with me had the biomedical engineering minor because they wish they could have had the major, and they come from like ME, EE, had a couple from BSc psychology. So kind of seeing it the other way. In my experience, I see a variety of different minors. We have a great nanoscience program here and neuroscience. So I think neuro and nanoscience are interesting minors that are outside of the especially the neuroscience minor is one that's outside of the general, STEM field. But having like an electrical or mechanical are probably the most straightforward that they have, especially if somebody knows that they say, I want to be in the biomedical, I want that to be my core, but I want to be, stronger in instrumentation or electronics. So then those make a lot of sense. But at the same time, like I said, neuroscience is an interesting one because I'll have a lot of students who really want to work on medical devices or therapies. Biomedical is the right fit for them, but something like a minor in psychology or a minor in neuroscience fits where they want to go work on neurological disorders, and this gives them that synergy as they look to grad school. And the nice thing with the biomedical engineering major is you can go a lot of different directions because you're exposed to so many different areas. You're kind of like at this intersection of all these different fields. So if you kind of know in advance, I think there's a lot of ways you could pair it with different types of minors. But I apologize, I don't have those stats. But if you for instance, email like our undergraduate coordinators, they can you'll find their information online. Some of the more academic coordinators would get you better information on that. And I think it's just again a question of interest. And I think biomedical, we get a lot of students who have a lot of different interests, which is why they like biomedical, because it has applications towards the medical field, but it really integrates. I should have mentioned chemistry. I think chemistry is, also chemical engineering is also a really, natural one to pair, especially if you want to go into more of the biomedical pharmaceutical side of things. Having that chemistry background is extremely valuable. So, but yeah, it tends to be like, where do you want this added expertise? And I see that a lot of people going to grad school in a different area, whether it's coming to biomedical from those fields or vice versa, for a master's or a PhD, to kind of give them that added more focused expertise in these two areas. And there may be something with more of the anatomy physiology. I'd be interested in one question, if you're interested in that, is for students who are on the BME more pre-med track. They may have more of the biology as their minor. That whether they get the official minor or not, there's a few classes like biochem and organic chemistry and some of the anatomy physiology that you kind of get through our program, but sometimes you have a couple add-on courses that you'll want to take for medical school requirements. Yeah, so if you look at our curriculum, it'll walk you through the concentrations within BME, but basically you can think of, we have a big focus on things like medical devices, entrepreneurship, bioinstrumentation and bioelectronics. Biomechanics is another one. So focusing on, there's really two types of biomechanics. There's tissue biomechanics, which is on as the name suggests, the tissue scale, and whole body biomechanics. We have a lot of really interesting work in our department. If you look up like the Helmet Lab or some of the work that Dr. Robin Queen's group does on movement biomechanics and whole body biomechanics. So that's a big one. Biomaterials, so both like for tissue engineering based biomaterials as well as like traditional biomaterials. Those are kind of so biomaterials, bioelectronics, bioinstrumentation, biomechanics. I forget which ones I've said, but those are the kind of the main areas where it breaks up into. The medical devices, biomedical entrepreneurship is something that we're at Virginia Tech really have been pushing. It's something that I care a lot about. I mentor a group of undergraduate students called Bioactivity, and it's a highly interdisciplinary group of students that's really focused on, it's kind of like doing undergraduate research, but it's more student driven and really focused on design of new devices and technologies that they kind of come up with, and really focusing on the entrepreneurship space. And that's in partnership with our business school here. And I've really enjoyed that a lot. And that's something that we do design pitch competitions. A former professor, Dr. Mark Van Dyke, and a current professor, Rob Gordy, run this entrepreneurship course through our interdisciplinary graduate program, TBMH, Translational Biology, Medicine, and Health. And they'll typically have an undergraduate engineering student, a graduate engineering student, a TBMH student, and I think a business student on these teams. It's like a semester-long competition. I think is something like the winners got like $5,000 that they just get to keep. I may have that number wrong, but it's a lot of fun competition. The teams come up with some really good ideas and get to go through this whole process of FDA regulation, what it takes to really get a device into the clinic, or start a new company and do the business model. And so that's something that I think will only expand in our coming years as our program grows. I appreciate you all listening to me. Again, reach out to me, and hopefully I'll cross paths with you down the road. But no matter what, if there's ever anything I can do to point you in the right direction or answer more of your questions, I'm always happy to do it. And I appreciate you taking the time to listen to me, especially my lab tour and mask. I prefer to do those in person, but we're all changing this year, and I think it's good that we're doing that and still making it work and still getting the opportunity to have these type of webinars. And hopefully this helped a little bit, give you at least an idea of what we do in our lab, and at least kind of an example of what a biomedical lab is. And like I said, a lot of great people in our department, a lot of interesting research going on that I'm happy to connect you with if you have specific interests. So like I said, feel free to reach out to me. Otherwise, thanks for taking the time today.